Hi friends, welcome back to my channel. So today let's see Voet and Voet textual book. Okay, now without any delay, let's start the class. Now without any delay, let's begin with the class. The traditional method of taxonomy, the science of biological classification, which are based largely on the anatomical comparisons of both contemporary and fossil organisms, are essentially incapable to prokaryotes. So as we know, the traditional method of taxonomy is based largely on anatomical comparisons. Okay, both contemporary and fossil organisms are essentially incapable to prokaryotes. Okay? This is because the relatively simple cell structures of prokaryotes, including those of ancient bacteria, as revealed by their microfossils remnants, provide little indication of their phylogenetic relationships. So, if we see the simple cell structure of a prokaryotic, so these prokaryotes are ancient bacteria, which is revealed by microfossils remnants. Okay, and they are providing little indication of the phylogenetic relationships. Okay. Phylogenesis, evolutionary development. So phylogenesis of the bacteria prokaryote is evolutionary development. Compounding this problem is the observation that prokaryotes exhibit little correlation between form and metabolic functions. So compounding this problem about prokaryotes, which is exhibiting little correlation between form and metabolic functions. Moreover, the eukaryotic definitions of a species as a population that can interbreed is meaningless for the asexual reproducing prokaryotes. So, eukaryotic definition of a species of a population can interbreed is meaningless, okay, for asexual reproducing prokaryotes. Consequently, the conventional prokaryotic classification schemes are rather arbitrary and lack the implied evolutionary relationships of the eukaryotic classification schemes. Section 1 to B. So, the conveying prokaryotic classification schemes are arbitrary, okay, and they are lacking the implied evolutionary relationships of eukaryotic classifications. The most widely used prokaryotic classification scheme, the Prokaryote, also known as Monera. So the Prokaryote means you can also know it as a Monera, have two divisions, the cyanobacteria and the bacteria. So these Prokaryote have two divisions, one is cyanobacteria and the other is bacteria. Okay. The latter are further subdivided into 19 parts based on their various distinguishing characteristics, most notably cell structure, metabolic behavior and staining properties. So these bacteria are further subdivided into 19 parts based on their various distinguishing characteristics. Okay. Most notably cell structure, metabolic behavior and staining properties. A simple classification scheme which is based on cell wall properties distinguishes three major types of prokaryotes: the mycoplasma, the gram-positive bacteria and the gram-negative bacteria. Okay, so this simple classification scheme based on cell wall properties, which is distinguishes three major types of prokaryotes. One is mycoplasma, another one is gram-positive bacteria, and the other is gram-negative bacteria. Mycoplasmas lack the rigid cell wall of the other prokaryotes. So as we know that mycoplasmas lacking the rigid cell wall of other prokaryotes. Okay, they are the smallest of all living cells, as small as 0.12 micrometers in diameter. So as we know that mycoplasmas are smallest of all living cells. Okay, they are as small as 0.12 micrometers in diameters and possess approximately 20% of the DNA of an E. coli. So mycoplasmas are possessing approximately 20% of the DNA of an E. coli. Okay. Presumably, this quantity of genetic information approaches the minimum amount necessary to specify the essential metabolic machinery required for cellular life. So, the quantity of a genetic material which is approaching the minimum amount is to specify the essential metabolic machinery required for cellular life. Okay. Gram-positive and gram-negative bacteria are distinguished according to whether or not to take up gram stain. So, the gram-positive and gram-negative bacteria are distinguished further by taking up of gram staining. Okay. The procedure developed in 1884 by Christian Gram, in which heat-fixed cells are successively treated with the dye crystal violet and iodine and then destained with either ethanol or acetone. So in the year 1884, Christian Gram, scientist, he have heat-fixed the cells successively treated with the dye crystal violet and iodine and then he destained with either ethanol or acetone. So for gram-negative bacteria possessing a complex outer membrane that surrounds their cell wall and exclude gram stain, whereas gram-positive bacteria lack such a membrane. So in this case, we found through this gram staining that gram-negative bacteria is possessing a complex outer membrane, okay, which is surrounded their cell wall and excludes gram stains. Whereas in gram-positive, the outer membrane is lacking in the case of the gram-positive bacteria. Okay. The development in recent decades of techniques for determining amino acid sequence in proteins. So the development in recent decades of techniques for determining amino acid sequences in proteins, section 7.1, and base sequence in nucleic acid, section 7.2a, has provided abundant indications as to the genealogical relationships between organisms. So in these techniques, they have known about the amino acid sequences in proteins. Indeed, these techniques make it possible to place these relationships on quantitative basis and thus to construct a phylogenetic based classification system of prokaryotes. So techniques which is making possible to place the relationships on a quantitative basis and it is constructing a phylogenetic based classification system for prokaryotes. Okay. By the analysis of ribosomal RNA sequences, Carl Woese showed that a group of prokaryotes he named the archaea. So, scientists, the analysis of ribosomal RNA sequences, Carl Woese showed that group of prokaryotes has been named as an archaea. Okay. Also known as archaebacteria, are as distantly related to the other prokaryotes. So, these archaebacteria are distantly related to the other prokaryotes also. The bacteria also called as eubacteria. So that particular type of bacteria is also known as a eubacteria, as both of these groups are to the eukarya, the eukaryotes. So if both the groups are present, then that particular bacteria we can call it as a eukarya or eukaryotes. The archaea initially appeared to constitute 3 different kinds of unusual organisms. So in archaea, initially there are 3 different kinds of unusual organisms are appeared. Okay. The methanogens, obligate anaerobes that produce methane, marsh gas by the reduction of carbon dioxide with the hydrogen. So 3 different kinds of unusual organisms. First one is the methanogens, where the obligate anaerobes that producing the methane gas by the reduction of carbon dioxide with the hydrogen gas. The halobacteria, which can live only in concentrated brine solutions greater than 2 mole NaCl, and certain thermoacidophiles organisms that inhabit acidic hot springs approximate 90 degrees Celsius and pH is less than 2. So halobacteria means it lives only in the concentrated brine solutions. Okay, and certain thermoacidophiles organisms which are inhibiting acidic hot springs. Okay, so they are living in the very acidic hot springs. However, recent evidence indicates that approximately 40% of the microorganisms in the oceans are archaea, and hence they may be the most common form of life on Earth. So, approximately 40% of the microorganisms in the oceans are archaea, and hence they are most commonly form of life on Earth. Okay. On the basis of a number of fundamental biochemical traits that differ among the archaea, the bacteria, and the eukarya, but they are common within each group. Who's proposed that these groups of organisms constitute the three primary kingdoms or domains of evolutionary descent. So, on the basis of number of fundamental biochemical traits differ among the archaea, bacteria, and eubacteria. Okay, but they are common with each group. Within each group, okay, who's proposed? So, Carl Woese proposed that these groups of organisms constitute three primary kingdoms or domains of evolutionary descent, okay, rather than the traditional divisions into prokaryotes and eukaryotes. However, further sequence determinations have revealed that the eukarya share sequence similarities with the archaea that they do not share with the bacteria. So, the further sequence of determination has revealed that eukarya share sequences similarly with the archaea that they do not share with the bacteria. Evidently, the archaea and bacteria diverge from some simple predominant life forms following which the eukarya diverged from the archaea, as the phylogenetic tree in figure 1.4 indicates. So, here in figure 1.4, they have indicated the phylogenetic tree. Okay, so this is the phylogenetic tree of this family tree indicates the evolutionary relationships. So this phylogenetic tree indicates the evolutionary relationships among the three domains of life. One is bacteria, archaea, and eukarya. The root of the tree represents the last common ancestors of all life on the earth. So bacteria are purple bacteria, cyanobacteria, flavobacteria, gram-positive bacteria. Archaebacteria comes under halophiles, methanogens, methanococcus, thermoproteus. All these comes under the archaea. And eukarya means animals, fungi, plants, ciliates, flagella, microsporia, slime molds. This all comes under the eukarya. Okay friends, that's all for today's class. If you like my video, please kindly like, share and subscribe it with your friends. In the next class, we will be learning about the eukaryotes. Okay friends, thank you. Thanks for watching. We will meet in the next class. Bye bye.